difference between domain Archaea and its comparison with domain Eukarya and bacteria both. So we'll be comparing domain Archaea at the same time with both domain bacteria and domain Eukarya. Now, as you know that domain Archaea are also known as ancient bacteria. So they are also known as Archaebacteria or Archaea, or they are also known as ancient bacteria. And we'll be studying, comparing them with bacteria as well as Eukarya at the same time. Now, first of all, the very first important difference is the difference of cell membrane. In the cell membrane of Archaea bacteria, there is ether linkage, the presence of ether linkage between glycerol and fatty acids. You know that in cell membrane, it is composed of lipids and embedded proteins. And the structure of cell membrane comprises of an alcohol, which is glycerol, and it's bounded with fatty acids. Now, these are the units of fatty acids, isoprenoid units, and as you can see, this is the phosphate group, and it is here you can see alcohol. So between alcohol and the fatty acids, there is ether linkage. Now, what is ether linkage? Ether linkage is that one carbon combines with another carbon through oxygen. So the linkage between two carbons, both having towards having single bond here, single bond, and both the carbons are bounded to each other via oxygen. So here you can see carbon and carbon, carbon of alcohol, glycerol, and carbon of fatty acid. They are both bound to each other through oxygen, and this linkage is called ether linkage. So ether linkage, and the second important thing is that the fatty acids, they are branched. That is, they have side chains available. So they are not in a straight chain; they are branched. So isoprenoid units are branched. So the true differences we can see, the two features we can see in the cell membrane of Archaea are that is the fatty acid and the glycerol is bound through ether linkage, through an oxygen linkage, and that the isoprenoid units are branched. So fatty acids are branched. Over here, if you look at the cell membrane of bacteria and Eukarya, you can see that the isoprenoid units are not branched. There is no branch in isoprenoid units. Thus, this is the phosphate group, and this is glycerol. You can see glycerol alcohol, and the carbon of the fatty acid is bound with glycerol through the ester linkage. Now, what is ester linkage? Look over here. If the carbon is bound to the other carbon via C double bond O. This is ester linkage. So this is one carbon, one chain, and this is the other chain, and they are both bound to each other via COO, C double bond O and O. So you can see here is a double bond present. Now, if you have, obviously, we'll be having some concepts of chemistry as well. That which bond is more strong, single or double bond? Single bond is not is very strong in nature as compared to the double bond. The energy required to break a bond determines its strength, and to break a single bond, a greater amount of energy is required as compared to a double bond. So that is why the oils they have double bond and they are liquid at room temperature, while ghee or fats, they all have single bond in their structure and they are solid at room temperature. So here you can say that because Archaebacteria can survive very extreme conditions, the highly saline or highly high temperature conditions we were discussing in our previous class. Why? Because their cell membrane contains ether linkage, which is more strong. It has the presence of single bond as compared to bacteria and Eukarya. They have double bond presence and they have ester linkage in their cell membrane. So they are not, their cell membrane is not that strong as compared to the no peptidoglycan or cellulose. Whereas the cell wall of bacteria, it contains peptidoglycan, which is a specific polymer. Polymer means a complex molecule which is made up of the combination of various monomers. Various monomers are joined to make a polymer. So polymer of sugar and amino acids. While if Eukarya you see, then we have cellulose plants. The cell wall of plants is made up of cellulose, while the cell wall of fungi is made up of chitin. Chitin is another very tough sort of a compound present in the cell wall of fungi specifically. So there's a difference in the cell wall here. Archaea have no peptidoglycan, no cellulose, no chitin in their cell wall. They have pseudopeptidoglycans because they have the presence of distinct proteins and amino acids in their cell wall. Now, if you look at the metabolism, there is a difference in the metabolism of Archaea and Eukarya and bacteria also. That is, they are specifically, they show some very specific metabolism. That is, they can do methanogenesis. That is the production of methane gas, which is used as biofuel. Whereas no, bacteria cannot do that, nor eukarya can do that. Bacteria have other specific type of metabolism activities. They show, that is, they can show photosynthesis. Archaea cannot. They can do fermentation. In eukarya, you see more complex type of metabolism because they are eukaryotes. They show cellular respiration for energy generation, and they show photosynthesis in plants and algae. So this is the third difference between archaea and domain bacteria and nuclear. And if you look at the genetic differences are, that is that we go for the sequencing of the whole organism. The whole organism is sequenced, the DNA sequence, and then it is overlapped. When the sequence of archaea is overlapped with eukarya and bacteria, it's surprising. It's been shown that they are more closely related to eukarya as compared to bacteria. So they have resemblance in their evolutionary history. They are more closely related to eukarya. Genetic sequence overlapping more with eukarya shows close evolutionary relationships with eukarya as compared to uh, eukarya resemble archaea more than bacteria. I hope it is clear. Let's take a quick overview of the topic. Number one, the cell membrane of archaea is made up of fatty acids and obviously an alcohol glycerol, but the linkage between the fatty acids and alcohol is ether linkage, while in case of eukarya and bacteria the linkage is ester linkage. Ether linkage is most strong, and that is why they can survive harsh temperature conditions. The second reason is that they have branched fatty acids as compared to these bacteria. They don't have branched fatty acids, and the branching of fatty acids makes the cell membrane more complex. Cell wall has no cellulose, no chitin, no peptidoglycan. They have distinct polysaccharides and amino acids, so we call them that they have pseudo peptidoglycans. In case of metabolism, they show methanogenesis. With bacteria and nuclear do not show that specific type of metabolism, which is very important, that they can generate a gas, biofuel, methane gas. Genetic differences. Now, in case of genetics, is surprisingly a very important fact that they resemble eukarya more as compared to, instead of resembling bacteria, they resemble more eukarya. I hope it's clear.